All right, we'll get started now. Hello, everyone, and welcome to Ingenious Talks Online, Organic Biosensors and Point-of-Care Technologies for Rapid Diagnostics, Early Detection and Monitoring of Alzheimer's, Parkinson's, and more. Thank you for joining us today. My name is Laura Kilpatrick, and I'll be serving as the moderator for today's event. While we meet today on a virtual platform, we'd like to acknowledge the Algonquin Nation, whose traditional and unceded territory we are gathered upon today. We do this to reaffirm our commitment and responsibility in improving relationships between nations and to improving our own understanding of local Indigenous peoples and their cultures. Together, please consider how we are and can each, in our own way, try to move forward in a spirit of reconciliation and collaboration. Now we'll review a few event items. So this is a Zoom webinar, which means that all attendee microphones and videos have been disabled. However, you do have the option to ask a question by typing into the Q&A box on your screen. While questions will be answered at the conclusion of the presentation, we highly encourage you to type them as they come to mind into the Q&A box on your screen. So you do not need to wait until the question and answer period to submit your questions. We'll try to get to as many questions as possible, but please be mindful that with the high volume of questions, we cannot guarantee that all will be answered. So you may also use the chat box on your screen to talk amongst each other and share ideas. Finally, a brief survey will appear on screen once you exit the webinar, and if you do have the time to fill it out, we'd really appreciate your feedback, and all answers are anonymous. So now I'm pleased to introduce to you our speaker, Dr. Ravi Prakash. Dr. Prakash is an assistant professor in the Department of Electronics at Carleton University, where he oversees the Interdisciplinary Organic Sensors and Devices Lab, whose research initiatives sustain and support a strong cluster of undergraduate, graduate, and postdoctoral trainee activities in organic electronics and biopolymer materials, flexible electronic components for wearables and human device interfacing, and smart interfaces for environmental monitoring applications. Prior to joining Carleton, Dr. Prakash was an assistant professor with the Department of Electrical and Computer Engineering at Queen's University and an NSERC postdoctoral fellow at the Provincial Lab for Public Health in Alberta. Today, Dr. Prakash will share impactful outcomes achieved by his research team at Carleton, including the development of two novel oral biosensor technologies that can detect hormones and proteins in saliva and exhaled breath condensate. Rapid, cost-effective screening of these biomarkers is vital for early detection and monitoring in Alzheimer's, Parkinson's, Crohn's, and COPD. Thank you, Dr. Prakash, for joining us today, and now I will hand it over to you.
Thank you very much, Laura, for the kind introduction. I'd also like to thank the Faculty of Engineering and Design for inviting me and for giving me this opportunity to talk today about some of the research endeavors that we are working on at Carleton University. So let me share my screen. I hope everyone can see my screen at this point. As Laura pointed out, we are here today to talk about what our focus in organic biosensors and point-of-care technologies for rapid diagnostics, and we work on several aspects here. But I think I'll pivot today's talk on the possibility of using such point-of-care devices for early detection and health monitoring for neurodegenerative diseases such as Alzheimer's and Parkinson's. So welcome, everyone. I'm excited that you've been able to join me today for this chat. So all this happens in a physical laboratory situated at Carleton University. It's the Organic Sensors and Devices Lab. We set it up about 4.5 years ago when I joined Carleton University in September 2018. The focus was to expand and bring in additive manufacturing resources, which will allow for low-cost fabrication of cutting-edge wearable and point-of-care technologies. So so far we have acquired and we are now utilizing these setups in our research endeavors. This includes combinational printers, which can handle low temperature polymers, nanomaterial inks, high temperature polymers, all to create complex devices and sensors for a variety of applications. We also have capabilities to deposit thin film, both functional and structural, for implementation in these devices and systems. And the Organic Sensors and Devices Lab is well supported by our existing Carleton microfab and nanofabrication and characterization facilities. So in all, we offer an excellent training resources for graduate, undergraduate trainees who are interested in working in these domains, and we, access to these infrastructures allows us to partner with both industrial and nonprofit partners for some very exciting novel research activities. So with all this that we have, our ambition is primarily to work on organic, chemical, and biological senses. We have a strong emphasis, our program has a strong emphasis on non-invasive detection of proteins, nucleic acid, and volatile organic compounds. So as you can see, we can use polymer conductive inks to create precision microstructures. We can create structural geometries. We can use these setups to interface sensors to read out systems, like a system on board sort of configuration. Now, organic devices, as the name suggests, we are reliant on functional materials which are carbon-based. So organic semiconductors for the transistor sensors, polymer dielectrics, which are good electrical insulation characteristics and good mechanical stability. And for conductors, we use both polymer and metallic conductors. So these materials in tandem allow us to build the sensor architectures and the multiple material aspects at the small footprint without really heavily relying on like a clean room environment, which makes the process costs go up and also large area manufacturing more challenging. In terms of sensing materials, so our ambition is to look for specific target analytes, such as hormone proteins and other proteins, volatile compounds, electrolytes, etc. So obviously, we rely on biomolecules as sensing material. A common example would be aptamers, antibodies. We'll go in detail along the presentation. We also rely on structured nanoparticles as sensing material. So molecularly imprinted polymers are one example. Graphene-based materials are excellent in this category as well. And of course, the selection of material and the process is tailored by our ambition to create sustainable sensor technology, which can be manufactured at low temperature, offer biocompatibility, which is a key criteria for wearable and non-invasive technologies. And obviously, as I mentioned, we achieve this using a combination of advanced manufacturing techniques, which allow us to combine these materials in strategic ways to build these devices. Okay, so let's pivot from what we have in our lab to the landscape of neurodegenerative diseases. And hopefully I'll try to convince you about the niche and the needs for point-of-care technologies in this domain. So we'll focus on Alzheimer's and Parkinson's. So I'll provide a brief overview, go over the current diagnostic methods, its advantages, challenges, etc., and then move on to how we are looking to implement our non-invasive or minimally invasive point-of-care technologies in this domain. So the term neurodegenerative disease essentially refers to progressive degeneration of cells in the central and peripheral nervous system. These conditions often lead to cognitive decline, like memory loss, and improper motor function, such as tremors, handshaking, etc. The Alzheimer and Parkinson's disease are the two most common of neurodegenerative diseases that affect our aging population. And I'll qualify that term aging here to point out that we are essentially referring to people over 65 years. Among these, Alzheimer's accounts for approximately 80% of all dementia cases. So you can see why we want to pivot towards these two, in more specifically Alzheimer's and Parkinson's. Now, Parkinson's also results in some cognitive impairment, but predominantly it leads to manifestation of loss of motor function. Now, these conditions often have multiple pathways to pathogenesis. What I mean by that is there are many different factors which can contribute to neurodegenerative disorders such as AD and PD, which results in a normal brain becoming a brain with neurodegenerative disease. So loss of nervous cells, loss of that interaction between brain and the central nervous system inside our body. So there are several factors. There are obviously genetic factors involve mitochondrial dysfunction. And then there are factors which are external, such as accumulation of certain metal ions, reactive oxygen species generation. And these two in particular, which as you will see is the focus of our approach to detecting AD and PD pathogenesis, which is a production of neurotoxic oligomers. So, proteins aggregating to create toxic aggregates, and deposition of fibrils, for example, amyloid fibrils in fluids and cells where they don't belong. So the reason, again, to bring the socioeconomic angle here, in 2016, almost 1 million Canadians suffered from Alzheimer's and Parkinson's disease, and these are the ones which were diagnosed back then. Unfortunately, I couldn't find a more recent stat. As you know, like the last 3 years have been pretty turbulent for Canadian healthcare and global healthcare system. A lot of our flaws have been exposed, and a lot of other factors have been, on the back burners while we tried to tackle the pandemic. The projection back then was that by 2030, with our aging population and with the burden of Alzheimer's and Parkinson's, it was estimated that the annual healthcare cost will reach up to $16.6 billion in Canada. AD and PD also happen to be the top 3 contributors to direct healthcare cost in Ontario and across Canada. So some of the reasons behind these stats, these mind-boggling stats, is because early diagnosis is often inaccurate, because various neurological disorders manifest into the same or similar cognitive and motor function deficiencies. Also, no clinical tests are available for Parkinson's disease even now. Most of the testing that is available happens to be physical and neurophysical imaging based. For Alzheimer, there are clinical diagnoses available, requires spinal tap, CSF, access to CSF sample. I will show the complexity to that, and again, the reliance on neurophysical imaging, which is highly accurate, but its reliance on centralized testing facilities leads to the challenges towards repeated and early monitoring of these conditions. So let's look closely at some of the neuropathological changes. For both AD and PD, these neuropathological changes precede clinical symptoms or physical symptoms. For example, the well-known neuropathological change in Alzheimer's disease is accumulation of beta amyloid peptides and phosphorylated tau proteins in neural cells. So what I'm referring to here is these monomeric peptide and proteins, which are pivotal to functioning and signal translation in our central and peripheral nervous system, interaction of brain to body interfaces. They become toxic if they become polymerized or they aggregate. These aggregates can be detected in multiple ways. The current diagnostic method relies on measuring them in cerebrospinal fluid, CSF, using immunoassays. And similarly for Parkinson's, and that's why we kind of want to look at them collectively. The disease itself is often characterized by the loss of dopaminergic neuronal cells, also correlated to aggregation of alpha-synuclein, which is a protein, monomer protein available or accessible in neural cells, which performs brilliantly in its in monomeric form. But as it conforms and polymerizes, it can form aggregates such as fibrils, which can lead to formation of Lewy bodies inside these cells, especially peripheral neural cells, and that leads to cell death and disruption of the signal transmission pathways. So as you can see, there are similarities in pathogenesis, at least from the point of view of protein aggregation and modification. And this is something we want to focus on, because this is something that's well documented. The only problem is that at the moment, these aggregations are detected using either CSF immunoassays or using neurophysical imaging. Now, the CSF immunoassay approach is daunting, because it requires collection of cerebrospinal fluid, not, the most accessible biofluid in our body, perhaps the least accessible, one of the least accessible biofluids. It's only about 150 to 200 milliliter volume present in the body in the spinal region. And for a typical immunoassay, it's required that you collect about 15 to 30 milliliters. The process is highly invasive, requires specialized instruments and specialists to access, the spinal fluid between the third and fourth left vertebrae of the spinal cord system. It's painful. It leads to certain side effects, especially in elderly patients. And this is a common deterrent towards proactive testing. And while the fluid itself recuperates within days, just the painful nature and the reliance on centralized capture sample collection facilities makes it challenging for therapeutic and continuous health monitoring. The other approach to precisely detecting Alzheimer's and Parkinson's is to look at brain images. So you have brain MRI, positron emission tomography, CT scans, etc., where you use markers such as fluorodeoxide, glucose, FTG, amyloid tracers, and you're essentially looking at brain scans to see abnormalities and presence of aggregates. Brain imaging is the gold standard. It's the best way to detect and monitor the stages of these neurodegenerative diseases. It's also the best way to exclude AD and PD from other types of dementia and other with similar cognitive and motor function declines, which may not be as severe. But again, the challenge is that these setups have limited access. There's usually a wait list to do these kind of imaging. So if there were ways to do some early monitoring, some non-invasive testing as a way to point to the clinicians or specialists that, okay, these imaging should be prioritized compared to others, it will help streamline the neurophysical imaging resources and their availability to people who need it most. So from that perspective, I want to share non-invasive testing reality. If you aren't very aware of what non-invasive and point of care testing refers to, in the next few slides, I'll try to establish that. And also talk to you about alternative biofluids, which allow for such non-invasive tests. So this domain is not entirely new. It's been a few decades at least where several researchers, including us, have been looking at creating biochemical sensors which can be small footprint, portable, low cost, and can be used for rapid or early stage monitoring in minimally invasive ways. So the early emphasis was on kind of creating lab on chip functionalities. So small footprint devices which can collect samples or take collected samples, purify them, mix them with reagents, and then you slide the chip under a benchtop setup for detection. That evolved into what the current phase looks like, which is you have disposable sensor kits with portable microelectronic readers and analyzers, something that our point of care ready devices look like. And in parallel, there is emphasis on wearable sensors, which have wireless or NFC connectivity to apps for continuous monitoring and feedback for doses. And we'll look at a few examples of that. And the projected new frontier is implantable continuous health monitoring devices. Okay, so I know a lot of the slides here are very illustrative, but that's just because I want to show as many examples of these technologies. The pioneering one, of course, are the Abbott Freestyle, the Libera continuous glucose monitoring system, which uses a minimally invasive approach. It still does blood draw, but less painful, and then essentially a portable unit which allows for data collection from the sensor and essentially tells people about their insulin level. And similarly, you saw the Abbott iStat in the previous slide here. Comparably, you have the Siemens Healthineers Epoch blood gas analyzer, which can allows room temperature testing of collected blood sample for a variety of analytes, blood gases, electrolytes, and analytes such as glucose, lactose, creatinine, etc. And then you have more novel. So these are still blood-based. But then you have some novel tests which are becoming mainstream, like the use at home saliva test for HIV status monitoring. It's only recommended for therapeutic use. If someone is taking medication and wants to monitor their status, they can use this. Now, obviously, with COVID-19, a lot of saliva-based tests were approved by FDA for rapid testing of COVID-19. Primarily categorized in rapid antigen and rapid molecular. The rapid molecular, of course, being more sensitive. So this is an example of one of the first Lucira Health home rapid molecular tests for COVID-19. And this was a saliva-based test. And there are other emerging frontier technologies where you're looking at wearables for monitoring a variety of biomarkers in saliva. So non-invasive biofluids and testing in non-invasive biofluids is the present. It's not something that we are projecting in a futuristic way. it's happening right now, which makes our job easy to convince the general society, to the healthcare agencies, that what we are building will become mainstream through extensive testing and prototyping. So non-invasive biofluids are likable because blood and CSF, as you have seen, complex to collect, needs specialized instrument, phlebotomists, limited shelf life of samples. If you don't use it during that period, you basically have to chuck it. It's high risk of contamination. So alternatives are samples such as saliva, exhale breath, condensate. These are your oral non-invasive samples. Then you have dermal ISF, interstitial fluid, sweat, tear fluid, etc. The sweat, tear fluid, essentially consider them as a lossy electrolyte-rich sample, which can be used for monitoring a lot of electrolytes, such as chloride ion and other electrolytes. So obviously, the likability is that ease of access. You can someone can use simplistic kits to collect sample, then I can either ship it to a centralized lab for testing or use point-of-care technologies to test them eventually. Disadvantage, they do have lower concentration of most target analytes compared to blood, and they're prone to larger variability of analyte concentration, again, compared to blood. So these disadvantages have limited emerging point-of-care technologies that wanted to rely on these biofluids. But that's not the case as assays have evolved, sensor technologies have evolved, and sample collection methods have evolved. So focusing a little bit more on saliva, because saliva is going to be a point of emphasis in this talk. So there are many ways analytes end up in a saliva sample. Hormone proteins end up in saliva through passive diffusion. Other analytes end up in saliva from capillary and adjacent cells through active diffusion. So it contains an abundance of biomarkers. I mentioned the term free hormone hypothesis, which essentially in a layman's term refers to the fact that hormone functionalities are correlated to hormone, the free hormone that can move from the gland that it's secreted to different cells. So it ends up in saliva using these pathways, and its concentration often is a very good indicator of physiological condition of your body. So there are several saliva-based diagnostic approaches that are currently in consideration, including protein biomarkers for cancer, autoimmune diseases, cardiovascular diseases, and we are hoping to add neurodegenerative to the mainstream here. Several pharmaceutical drugs, minerals, electrolytes can also be monitored in saliva. A key challenge would be the saliva production and collection, and preventing adulteration of collected saliva sample. So let's focus on sampling then. Now, obviously, majority of us have done some sort of swab tests already. No, thank you, COVID-19. So we know that there are many ways saliva sample can be collected. Now, obviously, this has, as I mentioned, this has become somewhat mainstream with identification and validation of novel biomarkers, improved test accuracy and sensitivity. And these are just some of the few examples of different tests that are becoming mainstream in saliva. So typical collection can be done in a few simplistic steps. So rinse your mouth with water 3 to 4 times prior to collection, and then allow saliva to accumulate in the mouth floor. Takes a few minutes. Could take longer depending on the age group and also pre-existing conditions. But once you have accumulated some saliva in the mouth floor, essentially following repeated spit or drool, you can collect it in a graduated tube, or you can use a cheek swab. Just go in a circular motion for a while. There's a lot of guidance on collecting these samples, including Public Health Ontario. So you can collect these samples and then essentially, as I said, if you have a point of care test, you can transfer it to the point of care test kit, or you can ship it to a centralized lab for subsequent testing. In our work, our sample of interest is saliva supernatant. Now, there is some macro debris in saliva, as in other media. So even after nice rinse, you may still have some food debris in there. You may have other kind of particulate debris in saliva. So those need to be removed. So it can be done through a simple centrifugation. For all the results that we will show you in our saliva test, they will be in supernatant. So we have cleaned it up to remove precipitates, but it's not a complex pre-treatment step. And we are also investigating if it's a necessary pre-treatment step. The other exciting outcome is that the supernatant yield from the saliva collection is pretty comparable to plasma yield from blood draw. So you're not at loss of the volume, you are at loss of analyte concentration, however, because typical saliva analytic concentrations are 2 to 3 folds lower, if not more, than the blood-based analyte concentration. Another sample that looks very promising for oral point of care testing is the exhale breath. An exhale breath sample can be classified to contain 2 types of analytes. You have the VOCs, volatile organic compounds, which could be endogenous metabolites or exogenous compounds, like ketones, acetone, etc., or gases such as ammonia. And then you have respiratory droplets, so also known as EBCs. And now they can contain proteins, metabolites, viral DNA, RNA, cell-free, as well as viral particles. The concentration levels will be low, again, probably lower than salivary concentration. The collection is a bit challenging too, unless you are testing through a breath column where it's a sample at test is used in a panel. Collection can be a bit more daunting than collecting saliva sample. Hopefully, I've established the feasibility of relying on a non-invasive sample such as saliva and exhale breath to some extent. So in the next 20 slides, let me show you one of these emerging non-invasive sensors, which we have worked with for about 4 to 5 years. And we have developed it, modified, improved it, and tested it to a point where we believe it's competent enough to be deployed for extensive clinical testing and can be adopted to a variety of disease detection and health monitoring through saliva. So I'm talking about organic electrolyte-gated FET saliva sensors. If it looks a very complex term, I want you to focus on transistors. I think all of us know transistors. They have 5 decades or longer history. They exist in all kinds of electronic devices. We trust them. It's a very established name. When I say organic transistors, I essentially mean younger siblings of these conventional transistors, where the transistor action is reliant on not on inorganic materials like silicon, germanium, gallium arsenide, etc., but on organic semiconductors. Now, organic transistors themselves have been around for a few decades. We all have smartphones with fancy touch screens and smart TVs. They all use organic semiconductors like pentacene and others, and they use transparent conductive polymers as well. That's what makes them so cost effective. So there are materials and technologies that already exist. What we and others like us are trying to do is show their potency as biosensors. And these transistor structures can be made into biosensors in a variety of ways. Today, we want to focus on what is known as electrolyte-gated transistor biosensors. Conceptually, the easiest way to say this is we want to insert the biofluid in the transistor geometry. And by doing so, what we are hoping to show is that changes to electrolytic or certain analytic concentrations will result in change to the transistor outcome, which will be device current in most cases. And these changes will be accurate and correlatable to those analyte concentration, which makes them nice, functional, and highly accurate quantitative sensors. So as you can see in this diagram, drawn by one of my PhD candidates, Roslyn Massey, her PhD thesis contributed quite a lot of the work in organic electrolyte gated FIT. And she and her, we have worked on this specific technology for a while now, moving it from these rigid configurations with inorganic materials to flexible iterations to integrated sample handling, and now to integration to electronic readout systems. So quite a bit of work has gone in, which leads to some exciting show and tell. So this cross-section essentially shows an OEG FET assembly, where you have the gate, source, and drain, as any transistor will have, the organic semiconductor, which leads to the device functionality, the functional layer, which in this case is an aptamer, a specific aptamer, and then essentially assembled all together through a modular fabrication process. So what I mean by that is you can print layers directly on these surfaces, which are flexible Kapton surfaces, and then you can combine them at low temperature using chemical or low temperature thermochemical adhesion process. And the bioanalyte can be incorporated through the microfluidic channel. The biorecognition molecule can be incorporated through either covalent binding or UV-assisted surface functionalization. So the design is simple. Most of the fabrication is done in our own lab using one or maximum two of those printing equipments. So what we are trying to project towards is that, hey, they can be built and scaled using low cost single footprint setups, which will lead to scalability in manufacturing and process reliability. So the process itself, right now, it takes a little bit longer because we're doing it using a chemical adhesion process to bind the microchannel. The microchannel is necessary because that allows us to incorporate any kind of samples, in this case, saliva supernatant, into the device geometry. So with this sensor structure, we're looking at a lot of key analytes. Most of the proof of concept research that we have done for the past 3 to 4 years has been with cortisol, which is a steroid hormone, often known as a stress hormone, because it correlates very well to chronic and acute stress. It's used for diagnosing Cushing's disease, Addison's disease, as well as it's very trendy in current endeavors of wearable devices for meditation and for monitoring your body's healthy living, for example. So our cortisol OEG FETs relied on aptamers as a biorecognition molecule, and more recently, and this is why the talk is tailored towards neurodegenerative diseases. With help from our excellent collaborators here at Carleton University in Professor DeRosa's lab and others, we have started looking into detecting or monitoring alpha-synuclein in saliva supernatant and synthetic samples. So again, we have established that there's niche to monitoring alpha-synuclein. And we are now investigating a similar approach, panel OEG FET sensor approach, where we can detect, using an aptamer again, detect beta amyloid concentrations and hopefully other similar aggregating protein markers. So you see the commonality. we want to build OAG-fed sensors. We want to preferably rely on a type of biorecognition molecule. We call them aptamers. So a quick introduction, if you're not familiar with aptamers. These are single-stranded, small synthetic DNA molecules, excellent to be used as biorecognition agents. They can be synthesized in lab using a specialized process. I have a slide to show on that. The benefit is that they can be made very specific to a target region of a target analyte, such as a protein. They're more stable on surface than antibodies. Aptamers can be incorporated for simplistic binding assays, so they don't rely on complex enzymatic activities, so better than electrochemical sensor in that regard. And they can be functionalized on surfaces with ease, which is greatly advantageous for building the kind of devices that we have in mind. The selection process is complex. You work with an oligonucleotide library, a repeated process looking at which of oligonucleotides bind selectively to target molecule using an affinity column. Then you elute it, you enrich it using nucleic acid amplification like PCR, and then you repeat the process. And then a lot of optimization happens using analytical techniques to select some of the best candidates, and then you go and test them in lab and then incorporate in sensor architectures. So the OEG-fed aptasensor that we are using and reporting are for proprietary alpha-syn monitoring. Our aptamers created and designed in Professor Maria De Rosa's ladder research lab at Carleton University. And collectively, Professor De Rosa and Professor Matthew Houlihan in neuroscience, they also investigate therapeutic implementation of this alpha-syn aptamer in monitoring, as well as implementing them for either reversing some of the Parkinson's effects in neural cells, or monitoring it and mitigating, reducing future effects. So I mentioned these are transistors, so essentially means that we will be generating amperometric output. So the current response from the device has a correlation to concentration of a target analyte. In this case, in this plot, it's cortisol, varying levels of cortisol in saliva supernatant collected by a student in the lab. You can see that there is a correlation. It's an inverse correlation, which is nice. So essentially, with lower concentration, our device is sensitive. We also have a nice log linear range for this sensitivity. It's a broad range sensitivity, which is great for a non-invasive point of care technology. And this correlation can be used to extract other meta parameters, for example, capacitance, cell capacitance, which is the system's way of doing sanity check that, okay, the data that we are collecting is real data. So we collect other parameters which corroborate that. The range is excellent. You can see that it's sensitive and specific, so it doesn't interact with non-specific analytes. And for cortisol, we tested in synthetic samples and in saliva samples to establish the detection range and compare it to the physiological range of cortisol in saliva samples. Now, since we implemented this technology for detecting alpha-synuclein, most of the next data is very recent. We're actually just working with Professor De Rosa and Professor Houlihan's team on submitting a manuscript with some of these. So they are very new data, but they're exciting data to show and present here. So what you see is unfiltered IV plots, current versus voltage plot of this amperometric sensor. But you can see that you have a broad linear range where you can focus the device to operate here. And essentially, you can see that the device delineates different analyte concentrations quite accurately in this range. And then you can see filtered data, which shows that you can modulate the amount of device current. This is a sort of a electronic integration parameter. So how much current gives us reliable delta V for optimized readout and for reliable broad range sensitivity. So you can see that it's quite well correlated, and the performance is excellent. The signal to noise ratio is also quite high. The filter data establishes that. And if we were to summarize this to show what we are seeing, see on a linear plot, you have a nice logarithmic dependence, which is great because that means that a log plot or a semi-log plot, it's a linear response. Transistors are great at doing that over a broad range of concentration. So you see that our APTA sensor can detect alpha-synuclein in synthetic samples. You also see that we can detect alpha-synuclein in saliva supernatant sample. Now, since the aptamer is designed to be specific to alpha-syn monomer, we also have demonstrated that it doesn't interact with even similar synuclein proteins, which are not alpha type. So it's very selective to binding to alpha-syn monomer, which is great because it allows us to kind of look at the total picture and say how much of alpha-synuclein monomer has oligomerized or has become fibrils. So this is great because it shows strong inverse concentration to presence of alpha-syn monomer, and it also allows us to create correlation plots for total to oligomeric concentration of alpha-synuclein, which is a very important parameter that can be monitored for detecting Parkinson's. So just to reemphasize, we have selectivity. So the device is sensitive to alpha-synuclein, but it's not sensitive if it's not selective if you are looking at alpha-synuclein, but the aptamer is not present on the device. Okay, so that's the selectivity from the aptamer. We also have specificity to the analyte. So with the same apta sensor, with the proprietary aptamers, you see that it's not sensitive to anything else in both scenarios, which means that we can rely on a device like this to separate and to quantify the total alpha-syn mono to total concentration in a large linear detection range, which covers the physiological detection range of alpha-syn in saliva and CSF. The limit of detection that we have seen so far comes down to close to less than 10 femtograms per liter. Can be extended further with sensor improvements. This is something that we constantly work on. And on advice from Professor DeRosa and her colleagues, to show the clinical relevance, the viability, we did recovery experiments, which is to say that we extracted calibration plots from multiple device testing. We collected anonymized saliva samples, which were spiked with different concentrations of alpha-syn. And then we tested them and tried to predict their concentration. And as you can see, we were able to recover the alpha-syn concentration in an anonymized sample with very high accuracy, close to 90%. We have also done another sanity check, so binding affinity quantification to show that the aptamer the aptamer binding on surface binding affinity is comparable to aptamer binding affinity in solution, which we believe is the case. A lot of the testing we do using benchtop setups because that allows us to collect good data. But in parallel, we also constantly create integrated footprints so these apta sensors can be deployed in portable configurations. This is one of those examples where two apta sensors are integrated on a system on board setup It's a low power setup, doesn't require a lot of voltage, fast detection. Interfacing of the sensor to the board is done using additive printing, and we have incorporated peripheral sensors to detect other peripheral parameters. So a block diagram showing the whole process. So you additively manufacture, then you test on benchtop, then you test on in the integrated setup to see if it's viable for clinical implementations. And then this is what we have worked on and established over the past couple of years. And we have done side-by-side sensor cluster designs and integration. So you can test multiple analytes in parallel or in sequence. To summarize, what we have shown is point of care ready. Not to say that we have done clinical trials, but we have shown that we are ready to do clinical trials, and we are ready to point towards the future of these devices for non-invasive testing. We can say that because we have shown detection range, which covers physiological range of alpha-syn in saliva, CSF. We have demonstrated, predictably demonstrated a low limit of detection, which is better than immunoassays. We have shown selectivity in detecting monomer to the aggregate. Our recovery experiments have been impressive. Granted, we haven't done a lot of them, but the plan is to do a lot of these to establish this. The reason we feel excited about this is even as we improve the sensor, we see the laboratory shelf life is amazing. It is semi-hermetically sealed, and we see sensor performance quantifiable on the upwards of 3 weeks. We have shown integration towards point of care footprint. We have shown multiplexing, not necessarily to the AD biomarkers, but this is what we are emphasizing on this year. And then essentially, we feel this is feasible because it offers low risk, high reward choices for repeat testing and continuous monitoring in aging population. So I know we are pressed for time, so I won't go into some of the other sensors in great detail, but I did want to point out that our lab works extensively on rapid nucleic acid testing in saliva, where we do sample extraction using a variety of methods. We have used droplet microfluidics to do DNA amplification from saliva and other samples. More recently, with a partner from Europe, we have looked at creating a rapid DNA test, which can be done at a constant temperature, because that miniaturizes the footprint, makes the devices faster, more sensitive to a variety of analytes. And this is something that we submitted late last year, where we investigated its viability in a variety of saliva, nasopharyngeal swab samples, as well as some environmental samples provided to us by the Cube Research Cluster here in Ontario. And to show the viability that, we have looked at other type of biorecognition molecules and implemented them for exhale breath testing. So just a quick preview, using molecularly imprinted polymers, binded on to simple microelectrode structure, we can detect high impact COPD biomarker, oxidative stress biomarkers such as 8-isoprostane. And we have shown to interface this with an aerosolized channel to collect breath sample and essentially quantify presence of these markers. So I'll summarize by saying that we are very busy working on novel point of care technologies, as well as implementing them in point of care ready configurations. I'm grateful for the amazing research team at our disposal. These are just the current students. Not all of them are there. Sorry, we are tardy at taking group pictures. And there are several others who have contributed to these projects and are now working in local industry as well as in other universities in the US. So I'll acknowledge our graduate team. I'll also acknowledge our research staff, support staff in Carleton University Microfab and Nanofab, funding agencies, acquiring these expensive infrastructure, relies on their support, and only then we can do these kind of novel cutting edge research. So all of them have helped acquire and produce these results. I'd like to thank all of our research partners, more specifically Professor Maria De Rosa in chemistry, Matthew Houlihan, Department of Neuroscience, and others whom we have projects related to health research, but may not be directly related to non-invasive point of care testing. So our industry partners, some of them that we have worked, we are currently working with for novel point of care sensors, as well as for therapeutic applications. Thank you very much for listening. And sorry, I think I went a little bit over time.
Thank you so much, Dr. Prakash, for that presentation. Yeah, we're almost at the end, but hopefully we have time to touch on one question. For those questions we don't answer right now, those will be forwarded to Dr. Prakash for review. So just one question here. So in terms of timelines, when might we see these technologies integrated into regular testing practices in Canada?
And that's a great question, and I know that it resonates with a lot of us. I would say that, you know, a few years ago, thinking of saliva-based point of care testing would have been far-fetched. But if COVID-19 has shown us one thing, that's the necessity to promote and to validate, to approve more at-home tests. A lot of them are saliva-based tests. So if we have seen like tens and 50s of these tests approved by FDA for COVID-19, so we feel pretty confident that that narrative will continue, and we have learned our lesson not to rely on centralized testing too much. So our footprint seems pretty robust. We obviously want to do more clinical testing, work with some clinical partners for that. But we hope that in the next 3 to 5 years, we have technology readiness, which can move towards commercialization of this or similar saliva-based tests.
Excellent. Thank you. That's encouraging.
I also see a couple of questions on my screen. We have
Yeah.
a few minutes.
Sure.
Is it okay if I go over it? I can share my screen again if others can't see. So a question from Jill: you have shown that your tests are not affected by the device used, and will they, but how do you know that system is not affected by other substances in saliva? Great question, Jill. And again, this is a mainstream thing. We extensively look at making the device sensitive to only one type of analyte. In electrolyte-gated sensors such as ours, the bulk effects are screened out from the surface effect. And by surface effect, I mean the interaction between the recognition agent, which is aptamer or an antibody, to the analyte that's binding to that recognition agent. So as long as we have excellent binding between the aptamer and this analyte, it's pretty independent of what is happening in the rest of the electrolyte, similar to how amino acids would work. So we are confident that it will not be affected by other content, and we do test saliva supernatant to ensure that, and we'll continue to research into it to ensure that happens. What are different expertise in your studies? All of those, I would say. Our team is pretty interdisciplinary, a mix of biochemists, electronics engineers, biomedical engineers, mechanical, biomedical, and we are trying to continuously work with collaborators and partners from chemistry, biosciences, chemical engineering, so that we have the expertise to work on such an interdisciplinary topic. Sorry, I couldn't resist. My bad. Great questions.
Great, thank you. Yeah, and I think that's about all the time we have for today. So thank you, Dr. Prakash, for sharing all of that interesting information and research that you're conducting. Thank you to everyone for joining us today. So on your screen there, you can see some ways to stay connected with the Faculty of Engineering and Design online. Also, if you are interested, you can join our mailing list for Ingenious Talks, and we'll put that into the chat. As well, one last thing is, so once you exit this webinar, there will be a quick survey up on the screen. If you do have the time to fill that out, we'd really appreciate your feedback, and the answers are anonymous. So thank you for joining us today, and have a great rest of the day. And thank you, Dr. Prakash.
Thanks, Laura, and thanks everyone. Have a nice day. Bye now.